Okay, so the plan will be, today we want to finish talking about central dogma, we talk about the RNA transcription translation. Thursday, this will be a guest lecture by Scott Baldridge, and I offer 5 points, attendance points, so you should come in. November the 11th, we'll talk about lacto-operon. Okay, there is a lot of operon. I only talk about lacto-operon, and everyone else, all those things you can learn by yourself in the other classes. This will be reviewed November the 13th. November the 18th will be exam 3. Okay, so that's, then we have a final section of a couple of lectures about the final exam. So because we postponed a day previously, so we're going to keep doing that. So you can fill the form with Amy's recording, but I don't teach in the spring because I have other things more important things to do. Otherwise, I'm going to keep stuck here, so I'm not teaching in the spring. Just let you guys know. Okay. Matt Casson will teach, but Amy basically will be responsible for that. Okay. Very good. Let's talk about our class. We have lots of materials we're going to cover. So we're going to review real quick about, wait a second, let's maybe, that's better. We talk about the central dogma. We say this is the outline of this section, and we talk about DNA. We're going through replication, and the genetic information later on transferred to the RNA, we call transcription. And finally, the information from RNA is going to go to protein. And this is called translation. Now, the first going to forming is amino acids. Then later on, being through the construction by themselves, become a peptides, and later on become a protein. So I want to first say something. What is transcription? Transcription is still letter to letter. You basically get mRNA, the message RNA, and get a codon. And then what is a translation? Translation, this is a letter to protein. Or we say letter to protein units, amino acids. So that is called a translation. This is still letter to letter. And we'll talk about that. Think about this. If you're speaking English, I'm speaking Chinese, and I do the translation, which means from English to Chinese, that is called a translation. Okay, so there is a different concept between that. And we finish this here. We're going to talk about the transcription and also the translation today. So first of all, transcription. So transcription, what we are talking about is RNA. So before we go to detail, we want to spend a little bit of time talk about the difference between RNA and DNA. So let's talk about that. So first of all, what is DNA? This is deoxynucleic acids. What is RNA? That's ribosome nucleic acids. So from the structure points, and you can see they are basically all the same. This is the base. And you have H, you have H, OH, you have H, you have CH2. You go here, we just say it's a phosphate. Okay, here, the same thing. You have a ribose, you have a base, and then you have H, 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 OH, H, CH2, and go here, we just say you have a phosphate. So basically they are similar. What's the difference is some places I didn't draw. For RNA, in the 2' carbon, this is OH. And in the DNA, at the 2' carbon, that is H. So the O is gone. That is what it means by these deoxy. So this is the first difference between that. The second thing, what you need to know, is the base. There is a difference. So what are the difference? The base for DNA, everybody knows, is ATGC. And for RNA, it is AGC, but instead of you have a T, here you have a U. So from a structure standpoint, they are similar. So what is T? We talked about 6 carbon. You go here, you have NH, you go here, NH, then you go CH3, here, go here. Then you say, what is the U looks like? It's very similar. If you want to draw this, we draw 6 carbon, exactly the same. Lots of the places, go here, then NH. So NH, H. Now what's the difference? That is carbon number 5. 1, 2, 3, 4, 5. Instead of you have a methyl group, I have H only. So we say uracil compared to thymine is replace methyl group at 5 carbon in a ribose with a proton or with an H. And we say at an unimportant place, H is replaced with CH3, or the CH3 is replaced by a H. So that's a major difference. Now, when you see the structure looks like this, when you see it looks like that, you will feeling about this is relatively, we say, unstable. That's why you always give you a signal something going to end or something going to happen. Okay, now what else are the difference? DNA, we know it is a double bond. 3' to 5', 5' to 3', and we say like this. So anti-parallel, we learn about that. Double helix. So double bonds, RNA most of the time is single band. That's a third difference. What's the last one? DNA we call replication. Replication, there is proof reading, which means they can solve or in a certain level can solve fidelity issue. We talked about RNA, there is no way you do that. This is the one why later on you will have molecular disease or genetic-based disease, and we call mutation. So when you move to, if you made a mistake here, you transfer to here, that's it. I would say generally speaking, of course some bacteria, some virus, or some of the microorganisms, or some microorganisms, we don't know, there are some exceptions. But generally speaking, once you go here, the mistake is there, it's going to transfer, go to here. Which means the amino acid sequencing could be different. So that is something we want to say before we talk about the transcription. Now, when you talk about transcription, we're going to also talk about the RNA. There are 3 major different RNA we want to talk about in the next couple of hours. We have mRNA. That's called message RNA. The message RNA basically is a codon. Its carry-on is 3 letters codon for amino acids. We also have a tRNA, and this is called transfer RNA. And transfer RNA, you should always remember, this is transferring is anticodon. And the last one is we call rRNA. This means ribosome RNA. And ribosome RNA, it is a structure of ribosome. And basically, you have 16S rRNA, and you also have 23S rRNA. And this is the first one underneath 30S subunits, and the next one is underneath or inside 50S subunits. What the S means, the Vandenberg units, the measurements for the precipitation. So these are the things we need to be very clear about. These are, we say, a foundation or preliminary knowledge for when we talk about the transcription. Okay? So once we have a transcription, these knowledge, we want to talk what is transcription. Transcription said very straightforward. So I have a DNA double bonds, 3' to 5', 5' to 3'. Let's say I have a small sequencing, which is ATGC, GCTA, let's say. Okay, I'm going to ask you to write in. What is the mRNA sequence? Can you do that? That's exactly what the transcription going to happen. So, what are we going to do? You have two double bonds of the DNA, and based on gene complementary policy, you get this bond, TA, CG, CG, AT. That everybody knows. Now I'm going to write mRNA. Lots of people knows how to do it. Because based on the first band, we call it a coding band. It's going to be exactly the same as a coding band. The only difference is instead of I have he, I have you, the others are exactly the same. But is this correct? And you forgot one thing. How they wrong? That's important because that's corresponding to the other bands of the DNA. This is what we call template band. Now, in order to do this, we need an enzyme. And this long and long sequencing generated at mRNA, we also call it polymerization. Since we call it polymerization, then we need thinking about there's an enzyme can do that, also called polymerase. And very lucky we had one called RNA polymerase. The RNA polymerase is discovered by Roger Cronenberg. He's also Cronenberg's son. That was a 1994 Nobel Prize for discover the RNA polymerase. Now, RNA polymerase is a hollow enzyme. What means a hollow enzyme? We talked about. It has a coenzyme and a cofactor. So they have coenzyme and cofactor. Okay, what are the coenzyme? It is composed by alpha 2 alpha subunits, beta, beta prime, and omega. Now, what is a cofactor? Sigma 70. What does 70 means? 70,000 delta are large protein units, and that's called sigma factor. And this is a very important thing. We want to talk about that. So we have RNA polymerase. We had mRNA sequencing we already had. The transcription the most important thing happens is at the beginning. So when we talk about the DNA, what is the most important thing? Is all those enzymes, how they work. For the RNA transcription, the more important is how they started at the beginning. Because RNA polymerase is not just to randomly go there to do the work. They only recognize a conservative sequence. So the conservative sequence is on the DNA. So let's say the DNA. Whenever we initiate, we call it a +1. On the right here, we call downstream, left side, upstream. The initiate place we call it a +1. At the distance to the left, which means on the upstream is about 10 BP, 10 base pair. There is a conservative sequencing called TA, TA, AT. And right there, that is we call -10. We also call it ribonol box. Now, not only for that, on the upstream, we go down, we go up around 18 base pair. We have another conservative sequence called TA, GA, CA. You can see that. Or TT, GA, CA. This is called a -35. Now you can do a gene complementary AA, CTG, GT, and this is AT, AT, TA. This whole area is called promoter. This promoter is recognized by sigma 70, or we say recognized by sigma factor, and generally speaking, sigma 70. Once it's recognized, the RNA polymerase will be attached to it. Once the RNA polymerase is attaching to it, they're going to start to do transcription. That's what happened. So said very simple. What is the function of RNA polymerase, or DNA polymerase, RNA polymerase? Number one, unlinked double-stranded DNA. Number two, reading DNA frame. Number three, biosynthesis of message RNA. That's a key for transcription. Now, we have to mention is that this sigma 70 is a general sigma factor, a cofactor, to read RNA polymerase to recognize this conservative sequence because of promoter, they can attach each other to do the transcription. But in the certain circumstances, it will be changed. So we have a table for that. Sigma 70 The promoter looks like that. The gene transcript is mainly a device, which means generally the function. If the bacteria in a beef steak and you cook it, they're going to respond to the heat. At that moment, if the bacteria is heat shocked, and then they're going through transcription, their sigma factor is different. We call sigma 32. You can look at the slides. And their conservative sequence is different. And let's say in a SIM agar, I have a protease of agar acid, and I'm going to do a movement because they have a petrotrochus flagella. At that moment, if the bacteria is moving, their sigma factor is also different. We call it a sigma 28. So you can see the difference is not the same as sigma 70. So here it tells you, every time the bacteria in their life cycle, if they are trying to do a specific job, their RNA polymerase is different. Which means their sigma, because their sigma factor is different. And the sigma factor is different when they recognize the promoter is also different. So they corresponding to each other with the enzyme has a conservative sequence for that. We don't want you to remember all these detailed sequences, but you absolutely need to know the sigma 70 factor recognize a general universal conservative sequence is TATAT, we call it a -10, and TTGACA, which is we call it a -35, +1, and this whole area we call the promoter. And that is important. Because when I came back next week, Tuesday, when we talk about lacto-operon, we also going to mention about this promoter and the RNA polymerase connected with each other again. Okay, so that is something is important. We want to talk about that. So we go back to see the slides. This tells you what is RNA structure. And we already explained to you. Then we talk about what is transcription. And this gives you RNA polymerase, what it looks like. It's a hollow enzyme, we already said, and this gives you example, different type of the sigma factors, and this is transcription initiation, and this is transcription elongation. Elongation, basically, once they do, they have is a hybrid between DNA and RNA, and have a mRNA sequencing comes out, and you can say with a bubble is a temporary RNA and DNA hybrid formed. Now, how do we know it's going to be stopped to do the transcription? There are two things you're going to do. One thing, if you see stem loop structure comes out, it will be terminated. Or you see rich new area, that means the transcription will be stopped. That's what I just said. The uracil, the structure, kind of means it's not stable. It will be stopped. So that's the reason we need to know a little bit about the structure, how work it is. Okay? So that is transcription and how we do the termination. Now, sometimes in a specific different things, we also can see some raw factor. So could it be stem loop system, rich U area, or growth factor? Once you have these signals, which means the transcription has to be terminated. That's the slides, which is giving you a conclusion, how they initiated, how they do the DNA template to the reading frame, how the mRNA is synthesized, and how finally they separated and terminated. That gives you overview picture of what it is. Okay, so that's what we talk about for the transcription. So the next one we want to move on to is a translation. How are we going to talk about this one? We're also going to be using a lot of the chemistry drawing. We want to explain it to you what happened. First of all, we have this slide. So we going to talk about protein translation. So let's move here, move down. So the whole transcription gone. We need a whole blackboard talk about translation. So think about the translation. You already know in your other classes. So remind yourself what you learned. Translation, you will know the genetic codon, get to know a ribosome, and you get to know tRNA, all those things. What you learned certainly will help you, but we are going to be a little bit different compared to what we're learning about chemistry class in here. First thing first, this slide is from another standpoint, talk about central dogma. See that on the top is DNA. I draw it already. Coding strand, template strand. And then you have an mRNA. The mRNA, the sequence, is exactly the same compared to the coding strand of DNA. The only difference is instead of you have T, here you have U. And then you have amino acid sequencing comes out. It's a 3-letter codon representing one or more amino acids. And don't forget we have tRNA. Okay, so that's an overview picture. And also, this is from the central dogma standpoint. So we talk about the translation. So we already said translation is from a letter to an amino acid sequencing, which is a precursor of protein. So we wanted to, so therefore there is like a codon system. How you do the coding and then you decode it. This comes out with the codon system. How are we going to do it? So you have a codon system. This is called a 3-letter. You already know. So let's talk about from the beginning. Okay, let's say 3-letter have a 3 box. This is box number 1, box number 2, and box number 3. So my first question will be, theoretically, how many codon we have? Remember what's the codon we are using? A, U, C, G. So how to do this simple math? Theoretically, how many genetic codon we have? A, U, C, G could be in the box 1. A, U, C, G could also be in the box 2. And A, U, C, G could be also in the box 3. So how many possibility is in the box 1? It is 4. How many possibility in the box 2? It is 4. How many possibility in the box 3? It is also 4. So we multiply by them. 4 multiplied by 4 multiplied by 4 is equals 64. So theoretically, we should have 64 genetic code. But how many we have? We have 61. What this tells you? That means there is a 3-1 doesn't do anything from the amino acid sequence standpoint, but they do their work which is giving you a signal to stop the translation. We call it a stop code. So what are stop codes? UAA, UAG, and UGA. That's a stop code. Right there. And you always remember, we have an initiator. The codon is always AUG. But AUG is an amino acid, which is arginine. Okay, so you have to remember that's a major thing. This is the first question I want you to think, even if you learned already. Second, how many amino acids we have? You learned in your 20, your class, or right now you maybe say 21. So we have 20 amino acids. We say 20 to 21 amino acids. But how many genetic codon we have? We have 61, excluding the stop codes. What this tells you? This tells you one amino acids could be translated by more than one codon. We call it redundancy. What they do, then how they do it. It's very interesting. We care much about the position number 1. That must be correct. We also pay much attention to the position number 2. That must be correct. But we give you flexibility for the position number 3. That's called the wobble effects. So, for example, you can see CUU, CUC, CUA, and CUG is all represents leucine. Which means the codon or the code at position number 3, it doesn't matter. Sometimes, not always. This is important because bacteria during the centrodogma process, they more or less will have fidelity issue, which means they will make a mistake, which means they will cause mutation. But if mutation suddenly happens, or sometimes happens on the number 3 box, it doesn't matter. So we call it a nonsense mutation, or we say call it a silence mutation. We call it a silent mutation. Which means, although the 3 codes has been changed, but the meaning of the amino acids, it doesn't change at all. So that's really good, which gives you life a little bit flexibility. The same thing everybody likes. So that's what the wobble effects talks about. Now, before we talk about the translation, we want to also know the structure of a tRNA. So tRNA structure is very special. It looks like this. Very special. 5' to 3'. They have different arms. This is the most important one. Everybody knows it. Anti-codon arm. This is what we call T theta C arm. What this tells you? It's very interesting. If I give you a sequencing from amino acids, or give you a codon from a bacteria, how do you know it is a tRNA? If you read the sequence, you will see there are some of the base, it's unusual. let's say py, pu, t, theta, all those. If that happens, then most likely it is tRNA. And we also have the other things, we call this a variable arm. And this is called DR. Okay. And this is whole thing looks like glow leaf. So that's the structure of tRNA. So we said already so many. What is going to happen for the translation? So we want to talk about that. What's the translation? We rarely go. Translation, it initially happens, we have a formula for that. So we had amino acid. We had a tRNA. And we need using ATP, which is the money, and also rely on magnesium. We will generate amino acid tRNA and release phosphates have AMP, or sometimes ADP comes out. Now the interesting thing here you need to know is how they are constructed with each other as a tRNA. And this is something you need to know how to draw, okay? A ribose. We had the H, we had the base. This is all the same. 2' we have H, we have OH. We have H, we have OH. We have H, we have CH2, we have a phosphate. This is a tRNA. Kind of, but what's the difference? Goes here. That's tRNA. Just draw like this. That's tRNA. But what are we missing there? It's amino acids. Where they go? Goes here. That's a key. That's how it is important. Yes. As an amino acids. We'll talk about the amino acids in detail later, okay? That's amino acids. That's how it goes. How they carry on, how they go. Okay? Now next, we want to talk about how the translation goes. So first of all, like we talked about before, there is a location for translation to happen. What's that? It's ribosome. And the ribosome, you need to know, it is a very few machinery, cell machinery, in both prokaryotes and eukaryotes, which means for both bacteria and fungi, they all have ribosome. And what a ribosome looks like? In the bacteria, we call the whole is 70S, and you have 30S subunits and the 50S subunits. This is the basic knowledge. And underneath or inside the 30S subunits, you have 16S RNA, and at 50S subunits, you have a very important one, is 23S RNA. So that's the basic knowledge you need to know. And the second, you learned in your biochemistry class, the first amino acid to be started is always, it's methionine. What is methionine? That is methionine. That's methionine. That usually what we say is for human being animals. For bacteria, it is different. We say the first one is N prime, formal methionine, tRNA. So what's the difference between that? The structure basically is the same. The difference is that's tRNA right here. You have an aldehyde group. This becomes formula. It's very special in the bacteria cells. So it is different. The fungi we say, or human being we say, MET. For bacteria, we say FMET. That's something you also need to know. It's a difference. Okay? So we said so many things. So next one, we want to mention to you how they started to do the translation. We want to move to here, then to explain to you. On this part of the blackboard, I'm going to draw how they're coming. And on this side, we're going to write the chemical structure. So you're going to well understand how that goes. So very beginning, we had a subunit of ribosome is 30S. The 30S subunits originally is separated for 50S. And they are attaching by initial factor 3. So remember, everything when you started to do, you need an initial factor 1, 2, 3. Lots of the genetic procedures, they all need an initial factor. Here we need an initial factor 1, 2, 3, 2. So initial factor 3 is originally attached to the 30S subunits to let them separate. If you have something happens, this is gone, attached with IF1. Initial factor 1 goes in. Which means I gave you a signal, we're going to start to do something. Okay, once you started to do something, okay, I have the mRNA going to come in. But you have to be careful. Does not mean any mRNA can start to do the transcription. The way, the sequencing for mRNA to do transcription, they must also have some signal, which we call it a conservative sequencing. It is called AGG. This is called Shine Dagano sequence. An acronym is SD sequence. And the SD sequence, they must be recognized by 16S rRNA of the ribosome itself. Then they can start to do the translation. And once this is started to do, on the other side, you have an tRNA gonna come in. And the tRNA coming is not just by itself. So you have FMET on the top. And you carry these signals there. And you need something to bring you in. So you need a boat. And this boat is called elongation factor 2. It's like a carrier, like a boat to let you in. But you all know that when you go some places, you need to pay the tickets. Is that right? So how they do? You need to bring the money. That is GTP. Okay, then this guy come in. Of course, you have to hydrolyze. So become GDP, you have to pay and become phosphate. Okay, when this comes in, what we do? The 50S subunits goes in. Okay, that's very beginning. That's very beginning what they do. Is a compact. Then they started. Once they started to do, it looks like this first of all. Whenever you go inside, let's draw here. You're gonna go 3 locations. This 3 location is very easy to remember because it's called EPA. That is not the Environmental Protection Agency. There is different meaning for that. What does E means? Exit site. What does the P means? Transpeptidal site. What is A means? That's amino acyl binding site. So this location is already being fixed to let you do the work. So how are they going to do? So let's say you have 50S, you have 30S. Okay, I have something coming. That's EPA. Okay, I have the first thing coming, let's say. Already be here. Okay, I have one, which is basically like this. Like that, like this. Okay, I carry on FMET. Okay, I'm doing this. Then you go here. I have another one goes in. That is AA1. Okay, how they move? They move this direction. Expressed here is also 5' to 3'. So you always remember 5' to 3' for everything. For replication, transcription, and translation. Now, how they connected with each other? This is the tacking here. And let it be moving. So what's the next step looks like? So let's draw here. Okay. The same thing we had 3 box. E, P, A. This guy moves to here. That's an empty one. Okay, then you go P. What's a place for P looks like? You're gonna have, go here. So that's 5 prime. Then you have an AA1 under the MET is on the top. And then you're gonna come in another one. Okay, I have another TRNA gonna go in. Go here. This carries AA2. And then you go move this direction. What's the next one will happen? So let's draw here. We draw all the time, then you will be understanding. That is EPA. This is going to release. So you have this free available RNA is released already. That's a released one. That's gone. And they are moving here. How they move? You have this tRNA right here. You have AA2, AA2. You have AA1, and you also have MET is always on the top. Then you go to another one right here. That's AA3. And then become long and long. Okay? That's how they goes. Now, when you see that, it's like a cartoon movie. It's a high school level, what you understand. We need to know more about that. So we draw here to see what's exactly happening. So E, P, A, we have a 3 location. Okay, let's say something is on the P already. So we draw like this. I'll just use A. Represents a base. So we have H, we have H, we have OH, we have a H, or we usually like this. This will be C, O, C, let's say MET, okay? Or FMET. That is H, that is NH2. And on the other side here, you have CH2, you have O, you have a phosphate right here. Then you carry on is a tRNA. That's 5 prime, that's 3 prime. Okay, let's say something is at A, have the very similar stuff is going. So, let's just draw it briefly, okay? I have this guy coming in. Let's go here, let's say I have O1, let's say. Okay, then H2. Then we go here, with H, CH2, we have a phosphate, so just say have a phosphate at tRNA, okay? What happened? If you draw like that, what happened? The next one. This thing here, gonna attack it. You're gonna attack in there. So what happened? This released. This is gone. If you don't know, you can draw it briefly like this. This somehow like that. That's released. That's used to find another amino acid. Carry on the anticodon right here. coming. It's like some carrier. And then how about here? It's going to go here. When it goes here, what's happening? So I use a different color. When they go here, this moves, this goes. So what it looks like? This is A, H, H, OH. This is H. Go here. One H, NH. Now go here. You have now, of course, it's a phosphate, it's a TRNA. What is this called? Peptide bonds. How they created? How they combine together? They need transpeptidase. That is inside 23S RNA of 50S subunit of ribosome. That is the key. Okay. And they go this way. Now, you're going to have something going to say. You say you go this way. Now you say you go this way. This is something for all the textbook have a little disadvantage. When you go the cartoon movie picture, go to the chemical structure, you have to upside down. Because it's 2D, it's not 3D, you cannot do it. That's why the textbook looks like this. You have to be flipped it over. Okay, that's how you understand. But the key thing you need to know, 5 carbon area is for a phosphate carried by tRNA connected with the 3' units, the 3' side, and here 3' this unit is important, 3' carbon is important because you are connected with the peptides. And this peptide is going to go long and long and long and long. And you always remember the first one in fungi is methionine, and in bacteria is F-methionine. Okay? That's how the story goes. Now when it stopped, go back to what we talked. Stop codon. When you see stop codon, that's it. Just tells you stop, then you release. Of course you release, you need releasing factor. And when you become long and long, you need elongation factor. And sometimes with TU. And those things we don't going to talk in too much detail. You learn in your biochemistry class much more detail. I only have so many times, so we go very briefly talk about the amino acid, the translation, how we're going to do it. Not done yet. If you really don't understand, that's a cartoon movie style picture there. And I tell you one thing, all these things, the DNA translation, transcription, and RNA translation, transcription, even lateral operon, all those things, they are movie at YouTube channel you can watch. I could be very lazy. I just broadcasting that for an hour. You can watch an hour. But I think you're already undergraduate, so we're in a more advanced level. That's why we want to talk about these chemicals and one by one. Okay, last thing, we get a couple minutes. So lots of people say they want extra points. And we already said the glycolysis TCA cycle, which is aerobic respiration, the chemistry name and the chemical structure will be the extra points. So here we talk about the amino acids at the end. So let's talk about that. So this is the amino acids, a very typical amino acids. Everybody knows. So amino group, carboxy group, H, and all side chain. And we have, let's just say 20, okay, 20 amino acids. Their difference is this all side chain. Do you know all these all side chain? Let's try, okay? I have been talking almost an hour. I don't know whether I still remember them. I maybe remember most of that. If I don't remember, I'll just cheat it to look at the notes. Okay, so based on the difference of the all, we have a different category. Majority of them you should know. It's not difficult. The first group is called phospholipid. I can say phospholipid. It's unpolar. We call it unpolar hydrophilic. So hydrophobic, which means they are not going to dissolve in the water. The simplest one is glycine. Then the next one, a little bit difficult, is alanine. Okay, we go a little difficult. What is this guy? Valine. Okay, then we go a little difficult. Go here. This is called leucine. And leucine had an isomerase we call isoleucine. Okay? So isoleucine. Then the last one, we have to draw the whole picture because it is a circle. So we go here, H, CH2, 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 go here, it's proline. And some of them we already know, some of them we don't know. It's okay. So not done yet, we're going to have some more. But this one we learned. Phenylalanine. We had a phenyl group, but alanine, we had a phenyl group. That's called a phenylalanine. And then the next one we did in the lab, this one we did in the lab too, it's a little difficult. Two here, NH is right here. That's called tryptophan. And remember what it is called? That's called the indole. What's the reagent? Kovac. Okay, tryptophan. Then the next one, a little bit tricky. That is called serine. Oh, sorry, cysteine. Cysteine is very special because we have the first time have used a sulfide group comes out. Then the next one, everybody should know. This is a very important one. Is methionine. Okay, that is unpolar hydrophobic. The second one, it is polar hydrophobic. Is that right? Still not dissolved in the water, but it's become polar. So what we have? CH2OH, serine. Then we have CH3OH, serine. Okay, the next one is not difficult. What is this one? Asparagine. Okay, next one. CH2, C goes here. Glutamate. So sodium glutamate, you all like it. It's called make a salt and very tasty. What's the other one? This one, always remember methyl as a phenyl group. Tyrosine. Okay, goes here. You're almost done. Because the next one is easy. We have 5 more left. Polohydrophilic. Sorry, this should be hydrophilic. Wrong. Polohydrophilic. This is unpolohydrophilic. That's polohydrophilic, which means they're dissolved in the water. You have asparagine. Then you very simple. You have aspartic acid, negatively charged. You have glutamate. It's very simple. You go here. You have glutamic acid. Okay, last one. Last week. This little tricky. That's always histidine. Okay, next one. All the nutrition scientists you should know. Very long. Lysine. Where is the lysine? In the milk powder. This is we call epsilon amino acids. Looks like this is very long. Because you have a lysine, what happened? The milk powder will change to yellow color very often in the room temperature, called Maillard reaction. Okay, what's the last one? We're almost done. Miss one here. Okay. Arginine. Okay, how we remember these 3? There's a joke. You know that. So back in 1980s, the girls in the campus, they're wearing the longer boots and showing off and walking around, have leggers. So we say her leggers are basic. H is histidine, L is lysine, O is arginine. They are basic amino acids. So that's a joke. For her legs are basic. And these days we have to be careful about these terms. I use this for let you remember all those 3. And what you should do, this has to work a little bit. Because it's a phenyl group. A little bit difficult here, a little bit difficult here. All the others, it's not difficult at all. What we do, this 0.5 points, this 1 point. So, 20 amino acids multiplied by this, 30 points. So, why we do that? We don't want you to give up. This is why you did so bad in organic chemistry class. Because after exam 1, exam 2, you already gone. And nobody wants to study. Then very soon you're from C to D and D to F. We want everybody in the classroom have the opportunity could save you life. Could be still work hard. So that is extra points, including aerobic respiration. Even if you don't know, you can get 1.5 points here. You can get 1.5 points here. Is that right? You can get 1.5 points here. You get this one. You get those two. You get that. Forget about like these histidine. That's tough. Lysine is easy. A very long CH2. You at least 1, 2, 3, 4, 5, 6, 7. You can get 10 points, 10.5 points. It's already a lot. So think about that. If you spend in time, you still could do very well in the exam 3. Okay? So that's all we have to do.